Ladies and gentlemen, welcome to the 2013 Croonian Lecture. I'm Jean Thomas and I'm the Biological Secretary of the Royal Society. Before we begin, could I ask you to turn off your mobile phones, please, or put them on silent, because the lecture is being filmed and it'll be available to watch online from the Royal Society archive in due course. If there's a fire, as far as I know, there's no fire drill planned for tonight, so if the alarm goes off, it'll be the real thing, and we're asked to leave through that door or through the doors at the back. This is apparently a fake door, so no good hammering on that one to get out. So to the Croonian Lecture. The Croonian Lecture is the Royal Society's premier lecture in the biological sciences. It was first delivered in 1738. It was conceived by William Croon, one of the original fellows of the Society, and among the papers left on his death in 1684 were plans to endow two lectureships, one at the Royal Society and the other at the Royal College of Physicians. His widow later bequeathed the means to carry out the scheme and indicated that the bequest was for, I quote, the support of a lecture and illustrative experiment for the advancement of natural knowledge on local motion, or, conditionally, on such other subjects as, in the opinion of the President for the time being, should be most useful in promoting the objects for which the Royal Society was instituted. This year's Croonian lecturer is Professor Frances Ashcroft, Fellow of the Royal Society. Frances read Natural Sciences in Cambridge, graduating in 1974. She then went to Leicester to do a PhD with Peter Stanfield on calcium currents in insect muscle fibers. During that time, she went to UCLA and she worked there on inwardly rectifying potassium currents. She set up her own lab in 1982, I think, in Oxford, and she's remained there ever since, most recently as Royal Society Research Professor for 10 years and now as Professor of Physiology. Within a year of arriving in Oxford, Fran had discovered the ATP-dependent potassium channel, and this has formed the basis of her work, most of her work, for the last 30 years or so. And we'll be hearing about some of this today, and some of this formed the basis for the award of the Croonian Lecture. Nearly 10 years ago, together with Andrew Hattersley from Exeter, who was sitting in the audience here, just off a plane, she discovered that ATP-dependent potassium channel mutations caused neonatal diabetes, and with Andrew as the medical partner, went on to show that sulfonylurea drugs were effective therapies, and I know we're going to hear more about this evening. Franz told me I mustn't speak for very long because she needs the time, and I appreciate that. So, ladies and gentlemen, could I now present Professor Frances Ashcroft to give this year's 2013 Croonian Lecture. Frances.
Thank you very much, Jean, and thank you all for coming. And if you can't hear me, just shout out and I'll move closer to the microphone. So I'd like to begin by introducing you to this young man here, who when he was just a few months old, developed diabetes so severe that it required hospitalization, and he faced a lifetime of insulin injections. And it subsequently became clear that he was developing more slowly than other children, and he was very late to walk and talk. And it turns out that he has a mutation, that's a genetic defect, in the protein which I've been working on for much of my life, which is known as the K-ATP channel, and I'll explain what channels are in a minute. And understanding how this channel works has led to a new therapy for him and children like him, and conversely, understanding his disease has told us considerably more about how this channel works and what it does. So I'll begin by reminding you that your blood sugar level is very carefully controlled. If it falls too low, even just for a few minutes, then the brain is starved of fuel and you can quickly die. If it rises too high for too long, then you get all the complications associated with diabetes. And one of the key regulators of your blood sugar concentration is the hormone insulin. And every time you eat a Mars bar, your blood sugar level rises, and this stimulates the release of insulin from the beta cells of the pancreatic islets. And insulin then causes the uptake of sugar into muscle, liver, and fat, thereby restoring your blood sugar level to its resting concentration. And if there is not enough insulin for the body's requirements, then your blood sugar level will rise and you get diabetes. And this can happen either because of impaired insulin action in the tissues, often as a consequence of obesity, or because you don't have enough insulin that is released from the beta cells, because there's something wrong with the beta cells. And there are a number of different types of diabetes, which I just need to remind you of here. So type 1 diabetes is the one which is otherwise known as juvenile onset diabetes, childhood in onset, and the beta cells are destroyed by an autoimmune attack in a sort of form of cellular-friendly fire. And because they have no beta cells, these people require insulin all of their lives. And really, nothing I say today will be of much relevance to them, I'm afraid. Then there's type 2 diabetes, which is extremely common and increasing, and is caused by multiple genes, and is exacerbated also by lifestyle factors such as age and obesity. And I'll be returning to this later. But what I want to focus on primarily is, as you've already heard, neonatal diabetes, which is a very rare monogenic condition. It's caused by a mutation in a single gene, and 50% of cases are due to mutations in this protein on which I work. So insulin, of course, was discovered a very long time ago in 1922 by Best, Banting, Collip and MacLeod. And this is the dog Marjorie, which is the first animal to be kept alive by insulin injections. So you may ask the question, since we have insulin, why should we be interested in understanding insulin secretion and what goes wrong in diabetes? And the answer to that is because insulin is not a cure. And this very fit and healthy young man, which you see here, who you see here, has a wristband on him because he's just had a heart attack. And diabetes is unfortunately associated with a large number of complications. It comes with an increased risk of heart disease, of blindness. It's one of the commonest causes of non-traumatic amputation. Many of the people on the dialysis unit are there because of diabetes. And as those of you who have diabetes may know, the risk of complications is reduced by good control of your blood sugar level, but you can never be as perfect as the pancreatic beta cell itself. So when I first arrived in Oxford, I thought that it would be interesting to work on something which had a medical relevance and where my mother and the taxi drivers would understand why I was doing it, which was not the case with stick insect muscle fibers. And I decided that it would be interesting to try to understand how a rise in blood sugar gives rise to the release of insulin. And this is what we knew. We knew that when glucose is taken up by the beta cells, it's metabolized, that is, it's broken down, and that leads to electrical activity in the beta cell membrane. And this illustrates a recording of that electrical activity. This is the type of spikes that you see. But the key question was, how does metabolism lead to this electrical activity? And due to the work of other people, notably Jean-Claude Oncan, I had the idea that this might be due to an ion channel closing. So I need to tell you what an ion channel is, and these are tiny little pores that are found in the membrane of every one of our cells and those of every organism on Earth. And basically they do nothing more than open and close, as you see in this model here. And when they're open, ions like sodium and potassium and so on can move through them. And when they're closed, they can't. And I'm particularly interested in an ion channel which is permeable to potassium ions and which is shut when it binds ATP. But we didn't know that when we set out to look for it. So I set out to look for a channel, and I used a recently invented technique by two German scientists, Lehr and Sackmann, which involves bringing a glass pipette with a very fine tip down onto the surface of the cell. And the tip is so small that you can isolate a single ion channel in the patch of membrane under the electrode. And then we add glucose to the bath solution, and because the seal between the membrane and the glass wall of the pipette is so tight, glucose has to be taken up by the cell, so that it can be broken down and then act from the inside on the ion channel. And what I found when we did this was that there was a channel that was active in the absence of glucose, and that's shown here. For those of you who aren't familiar with these recordings, this is when the channel is closed, this quiet bit up here, and here it opens and closes many times. And when glucose was added, channel activity disappears, and it comes back again when the glucose was removed. And this, I have to say, was really, really exciting to me, and this is the sort of thing I dreamt of for years. And even more interesting, when glucose was tested in the presence of metabolic blockers, channel activity was not inhibited, which indicates that this is a channel which is regulated by glucose metabolism. So the question was, what was the metabolic product? And at almost exactly the same time as I discovered a channel that was regulated by glucose metabolism, Cook and Hales, working in Seattle, discovered a channel that was blocked by intracellular ATP in pancreatic beta cells. And what they did was to do the same type of recording but remove the pipette from the surface of the cell, so ripping out a small patch of membrane with its intracellular surface facing the bath solution. And then they added ATP on the inside. This is a breakdown product of glucose, and you can see that it closed the channel. So I, of course, showed to my own satisfaction that these were the same channels. In fact, you can already see that they look very similar. And I was all ready to submit my paper when I got one to review by this man, Patrick Roseman. And I was rather annoyed about this, as you can imagine, but of course I accepted the paper because it was a very good one. And a few months later, I met Patrick in a swimming pool in Alicante, as one does. And we decided it was much better to collaborate than to compete, and we've been doing so ever since, and he's here in the audience tonight. And so we very quickly then knew that in the pancreatic beta cells, when these potassium channels are open, there is no electrical activity and thus no insulin secretion. But when your blood sugar level goes up, glucose is taken up, broken down to ATP, closes the channels, electrical activity is initiated, and insulin is released. And Patrick, unfortunately, also was the first to show that sulfonylurea drugs, which had been used for about 50 years to treat type 2 diabetes, work by closing these channels and thereby stimulating electrical activity and insulin release. And it doesn't matter whether metabolism is low or high, they act completely independently. So you can see from this that the take-home message is that when the channels are open, no insulin is released, and when the channels are shut, insulin is released. So it doesn't take a genius to realize that if there is a mutation that results in the channels either not being present in the membrane or always being shut, then you'll have continuous electrical activity and persistent insulin secretion. And in fact, people will be born with too low blood sugar levels. On the other hand, if you have mutations that prevent the channel from closing when ATP levels rise, then electrical activity will not be initiated and there'll be no insulin release. So we set out to look for those mutations. And unfortunately, it took 20 years to find them. And the reason it took so long was because the channel is really rather complicated, and it's made from two different types of protein subunit. One of them, known as KIR 6.2, forms the pore, and 4 of them come together to form a pore with a hole through the middle through which the ions move. And each of them is associated with a much larger regulatory subunit, which binds sulfonylurea drugs and is known, therefore, as a sulfonylurea receptor. And both of these proteins are needed to make a functional channel, and both of them are required for the glucose-dependent regulation of channel activity, because ATP, this breakdown product of glucose metabolism, inhibits the channel by binding to the KIR 6.2 subunit, and on the other hand, magnesium ATP, so that's just ATP in the presence of magnesium, stimulates channel activity by interacting with the sulfonylurea receptor. So the activity level that you get inside the cell depends on the balance between this inhibition and this stimulatory effect. So once we had got the proteins and knew their DNA sequence, it was possible to screen patients' DNA for mutations in the channel. And we screened a large number of patients, and we didn't find any mutations at all, although we did find a common genetic variant that was associated with a small increase in the risk of the disease. And then one of the clever postdoctoral fellows who worked with me, Fiona Gribble, said, you know, Fran, we shouldn't really be looking at people who have type 2 diabetes. You only get that in later life. What we should really be looking at is people who are born with the disease. So I went back to my clinical colleagues and I said, could we have some DNA, please, from people who are born with diabetes? And they all shook their heads and they said, they don't exist. And I should have remembered the motto of the Royal Society, nullius in verba, take nobody's word for it. Because in fact, they do exist. And one person who's also in the audience who knew this was Andrew Hattersley from Exeter University. And Andrew went out and made a collection of the DNA from people who were born with diabetes or who developed it within the first few months of life. And this is in fact a very rare disease, around 1 in 200,000 people, and it's associated with very high blood sugar levels within the first 6 months of life, and very low birth weights because insulin is a growth factor. And 50% of these patients have mutations in the genes that code for our ion channel. And I have to say, I've never forgotten the day that Andrew rang me up and he said, I think you should sit down, Fran. I've got something to tell you that's rather exciting. So I sat down, and then he told me that they discovered the first mutation that caused neonatal diabetes, and it was in KIR 6.2, and he asked us to collaborate. And I should say that most of these mutations occur spontaneously. They're found in the eggs and sperm. So we soon became aware that in addition to the diabetes which these patients had, about 20% of them also had developmental problems. It took them a long time to learn to walk and talk, and they showed muscle weakness, and about 3% of them also had epilepsy. And Andrew and I had the privilege of naming a new syndrome, which we call DEN syndrome, for developmental delay epilepsy and neonatal diabetes. So the question is, how do these mutations cause neonatal diabetes? And I'd just like to show you where these mutations are found in the structure of KIR 6.2. So what we're looking at now is a three-dimensional model of the channel. We haven't got the crystal structure. We don't have the atomic structure of our channel yet. This is proving very difficult to get hold of. So this is based on related ion channels, including a bacterial one that we managed to crystallize together with Declan Doyle. And every amino acid which is mutated in neonatal diabetes is illustrated here in red. And these mutations were found by Sian Ellard working together with Andrew. And the model is with the help of Phil Stansfield and Joseph Haider in Mark Sanson's group. And I'll just show you what it looks like. So here's the membrane, and this bit of the part of the channel is all intracellular, and the purple are the potassium ions moving through the center of the hole, and what you're looking at is really a little bit of Hollywood because we're morphing between models of the open and closed state. And in purple here is the ATP, and you can see that the mutations largely congregate around the site where ATP binds, and also in parts of the channel that move when it opens and closes. So what do they do? So to address that question, what we did was we took the DNA that codes for the channel and injected it into Xenopus oocytes, into frog eggs, which act as very good little biochemical factories. They make lots of the protein, they insert it in the membrane of the cell, and then we can pull patches from them and add ATP to the inside of the membrane and see what happens. And what you see here is for the control, the wild-type channels, you'll see that ATP blocks the currents very nicely, but it has no effect upon the mutant channel. And so we plot the current in the presence of ATP is a fraction of its absence to get the dose inhibition curve, which you see here. And you'll see that at physiological levels of ATP, indicated by this purple bar, there's very little current for the wild-type channel. But for the mutation, there's a really large current. And this is a mutation that causes not only neonatal diabetes, but also epilepsy. And you can see the current is very large. And if we look at a mutation that only causes neonatal diabetes, but is found at the same amino acid residue, you'll see that the current is much smaller. And this turns out to be a common theme. The lower the ATP sensitivity, the more the ATP sensitivity is reduced, the less ATP blocks, the worse the clinical phenotype. So at the far end here, we have mutations that cause neonatal diabetes with epilepsy. Then we have the mutations that cause neonatal diabetes on their own. Then we have mutations that cause diabetes, but it occurs in around the patients when they're 20 or so. And at the very far end, we have a common polymorphism that's found in 58% of the population, so will certainly be in some of you here, that increases your risk of getting type 2 diabetes. So what was, to me, very exciting about these findings was not just the science and understanding how the channel works, but the fact that it had implications for the therapy of the patients. Because this disease was initially thought to be a very rare form of type 1 diabetes, which, as I've already told you, results in the total loss of the beta cells, and so that the patients require insulin all their lives. And so it was always treated by insulin injections. And as these young men say, life with diabetes, no easy feat. And some of the children don't exactly appreciate injecting themselves with insulin. And it's much more difficult for a newborn baby, of course. And what the work that Andrew and I had done showed was that, in fact, these mutations led to the channel being open all the time because it was insensitive to ATP. The consequence was there was no electrical activity and thereby no insulin release. So the beta cells were essentially switched off. And we already knew from Patrick's work that we had a drug that could block the channels and thereby turn on the beta cells again. The issue was, did these patients have any beta cells left? And did the drugs actually work on the mutant channels? So to address the latter issue, we looked at the efficacy of block by these sulfonylurea drugs. Here for the control channels, you can see they're 100% blocked at this concentration of drug. And here for many of the mutations that cause neonatal diabetes. And you'll notice that many of them lie above this threshold line. And what that threshold line indicates is that if patients are transferred, that the patients who have these mutations that lie above the line are able to transfer from insulin therapy to sulfonylurea therapy. And it was Andrew who figured out how to do the actual transfer. Because as you can imagine, this is no easy thing to take a person who's being kept alive on insulin and say to them, well, we think you might have some beta cells. And if you do have beta cells, it's probable they can be closed, that they have the channels open and they can be closed by this drug. And it will be much better for you if you take it. But in fact, it turned out that it wasn't such a risk because most patients respond to drug therapy and 90% of them have now transferred to drug therapy and both their clinical condition and quality of life improve. And this is work from Ewan Pearson working with Andrew, where you see the average blood sugar concentration over the previous 4 weeks or so on insulin and on sulfonylureas. You can see that the number comes down, in some cases into the normal range, and this is really wonderful because it means that the risk of them getting diabetic complications is less. And their blood sugar level is also much better controlled and they have fewer hypoglycemic attacks. And for the patients themselves, these are some of the things they say, that they can, their parents now let them go and play football and sleep overnight with their friends, so maybe sleep with their friends is not quite right, and that they feel normal. And for me, this was the most surprising one, when a young girl turned to me and said, thanks to you, I can wear a dress. And I said, what do you mean? And she said, well, I don't need a waistband to hang my insulin pump on. And it's something that, as a woman, wearing a dress to give a lecture, I always remember, because I never have anything to hang the microphone on, which is why I'm pinned to the desk. So what about the neurological problems? What about the developmental delay and muscle weakness and the attention deficit hyperactivity disorder that these children have? And just to give you an indication, this little boy is 2 years old, and normally at 2, kids are tearing around, but as you can see, he's not even able to crawl properly because he can't push himself up into his hands and knees because he doesn't have the strength. So the question is, what's going on? And it's because the K-ATP channels are not only found in the pancreatic beta cells, they're found throughout the body, as you can see here, in the brain, the heart, the skeletal muscle. And in all of these cases, they link the metabolism of the cell to its electrical activity. So we ask the question, what causes the muscle weakness? Is it a problem in the muscle, with the K-ATP channels in the muscle, or are the nerves not telling the muscles to contract? And to address this issue, we made a mouse model of the disease. Or rather, Christoph did. And we took the mutation, which this young man has, which of course is neonatal diabetes with neurological complications. And we used a clever, not designed by us, I have to say, but a clever genetic technique known as the Cre-Lox system, which enables you to turn the gene on in the tissue of choice at the time you want. So we turned it on either in muscle or in brain, and then we gave the mice weightlifting exercises. And here you see what happens when you turn the gene on in muscle or in nerve. The grey are the control animals. Here, when it's turned on in muscle, you see that there's no effect. And when we turn it on in nerve, you can see that there's a small reduction in the ability of the mice to lift weights. And also, they have impaired motor coordination, as you'll see here. So the mouse is placed on this pole, and it feels exposed. So it twizzles around and runs back along the pole to the bench. And you'll notice that the control animal is able to do this perfectly happily, but that the one which has the mutation is having great difficulty. Its feet are slipping, and it's going to fall off. And I can assure you that this doesn't harm the mouse. And what this tells us is that the motor problems, the difficulty in movement, are neuronal in origin. They occur in the brain. And I have to say, the same features, the same impaired coordination are also seen in the patients. So the question is, can we have a look at which bit of the brain that this is taking place in? So this is a map of a cross-section of the brain, and the darker bits show you where the channel is found, and you can see that it's in this bit of the brain which is known as the cerebellum, which is involved in movement control. So we looked at the nerve cells, or rather James looked at the nerve cells in this bit of the brain, and what you see here is electrical activity. Each of these spikes is really a spike of electrical activity in the nerve cells, and you'll see that in the control animals they're really quite active. But the frequency is much less in the animals that carry the mutation in their nerve cells. But importantly, we can recover normal electrical activity by bathing the cells in a sulfonylurea known as torbutamide, so it completely recovers. So the question then is it possible to recover the neurological, can you help the neurological problems in these patients with sulfonylurea drugs? I mean, obviously when they are on insulin won't do anything about the neurological problems, but the drugs should block both the beta cell channels and those in the nerves. If, that is, they're able to cross the blood-brain barrier, and if the K-ATP channels are not necessary during development, because if they're necessary during development, irreversible changes in the brain might have occurred. Well, I'm going to show you one little video of the same young man who you've seen previously, and after 1 month after the drug, you can see that he can now walk, and he's starting to talk. So this was incredibly exciting to us, but I would caution with two things. First of all, he's only one child, and secondly, he doesn't recover to the same level as his peers. But Roger Highfield wrote this story up in the Daily Telegraph. And the mother of this young boy read the story, and she thought, that sounds just like my son. He's 5 years old, he's only just started to walk, and he still can't talk. And so she got in touch with her doctors, and her doctors with Andrew, and he was given sulfonylurea drugs, and within the first 3 weeks of taking the drugs, he said his first 3 words. So all of this is very nice, and there are other anecdotal stories, but you can always say, well, it probably would have happened anyway, because I can assure you that no parent wants you or will allow you to try the experiment of taking the child off the drugs and seeing if they get worse again. So I think there is some evidence that the drug can get into the brain, and that comes from the work of our Polish colleagues, who first looked at this young girl. Before glibenclamide, she was hyperactive, but she fell down a great many times. She had difficulty sustaining attention. And 6 months after taking sulfonylureas, she was able to sit still. Her IQ had gone up, presumably because she was able to focus more easily, and she was able to attend normal school, and she no longer fell over. And this is what her brain scan, her SPECT scan, looked like before the drug and after the drug. And what you're looking at here is the brighter the red, what it means is the higher the region of blood flow. And it's believed that the blood flow indicates the level of nerve cell activity, and that the more activity you have, the more blood flow you'll have, because the nerves need more energy and oxygen in order to function. So what you'll notice is that this part of the brain is not perfused prior to taking the drug, and that after taking the drug, other regions of the brain, including the cerebellum, are improved in their perfusion. And this shows you the results from several children on insulin. So this is the signal intensity in the cerebellum on insulin and on sulfonylureas. And you'll see that in most cases, blood flow increases, suggesting that the drug is getting to the brain, blocking the KATP channels and stimulating electrical activity. With the exception of this mutation, and this is a mutation that does not cause neurological problems, and you can see that it starts high already and doesn't change. So the drug probably is getting into the brain. But actually, experiments that we've done very recently, or Caro and Holger have done very recently, suggest that it's actually very difficult to get the drug into the brain at high concentration. If we implant a subcutaneous pellet of glibenclamide, which releases the drug very slowly, you can see that there's a very high concentration to be found in the blood, but almost nothing in the CSF, which is another name for the fluid that bathes the brain cells. If we deliver it directly into the brain, then we can see it in the brain, but an awful lot arrives in the plasma. So it appears that it's very rapidly pumped out of the brain. And that may be why it's so difficult to get enough drug into the brain to treat these patients, and why they require very high doses, and probably to some extent could be the reason why it's not possible to completely reverse all their problems. And the other possibility is that it may be that there have been developmental changes and that the KATP channel was acquired early in life. Because we know, you just look at these two beautiful children, brother and sister, and he was started on the drug therapy at 4 years old, and she was started immediately after she was diagnosed. And she is doing very much better in terms of her neurological symptoms. So what I've told you is that neonatal diabetes mutations in either of the two subunits of the KATP channel impair the ability of ATP to close the channel. The greater the reduction in block, the larger the KATP current and the more severe the phenotype, and that many mutant channels can be closed by these drugs. And more than 90% of patients can transfer to drug therapy, and in all cases their blood sugar level is improved, and in all of those who can, all of the 90%, and that some improvement is also seen in the neurological problems. And I'm not sure how much it's really due to the muscles. I think certainly better muscle tone will help their speech as well as their walking. And this is a good example of pharmacogenomics, where the drug is tailored to an individual's genetic constitution. But I'd like to point out that it takes a very long time, and no matter how fast the final stages might appear, there's a lot of work that goes on before that, and it requires an enormous amount of investment. So, a huge amount of money. So, it doesn't work when the funding bodies say, you can do translational research in 5 years. It's far too short. So where do we go next? I'd just like to finish by saying a few things about type 2 diabetes, because we're undergoing a global epidemic. Currently, 336 million people are affected, and that is continuing to increase. It's around 10% of the US population, and it's really, really expensive. It consumes around 10% of the healthcare budget in most Western societies, and it costs the National Health Service £1 million an hour. That's something we can't afford, especially if it's going to increase. So what goes wrong in type 2 diabetes is certainly the case that insulin secretion is impaired. This is work from Patrick Rosman, and you can see that so what's happened here is islets have been isolated from organ donors, and you'll see that those from a non-diabetic organ donor increase their insulin secretion when you raise the blood glucose concentration, but this is not as effective in islets that are isolated from a type 2 diabetic. So the question is, why is insulin secretion impaired? And certainly part of that is genetic. And we still don't really understand what's going on here. We know that multiple genes are involved, including, as I've already told you, my favourite ones. But there are many, many more, and how they work together is still not understood, and something that we're all struggling with. But lifestyle factors are also very important, and I think this is beautifully illustrated by this index of newly diagnosed diabetes cases in Norway between 1925 and 1955, and you'll note the marked crash in incidence during the Second World War. And this, of course, is because people actually had a better diet then. Because the big problem that we have at the moment is the huge obesity epidemic, which is driving the diabetes epidemic. And the other problem, of course, is that diabetes is increased by age, and we're all getting older. Well, we obviously can't do much about getting older. That's sort of inevitable, at least at the moment, but perhaps we should think about trying to not get too fat. But it's not all bad news. Not all fat people will actually develop diabetes. This is the UK's fattest man, Paul Masson, who is now actually half this weight. But he did not have diabetes. So it's very important to point out that many people who are fat do not get diabetes, and it would be very good to understand what protects them from diabetes. And not all old people will develop diabetes either. And the other thing that our work has taught us is that beta cells remain functional even after years of diabetes. So that, in fact, as in this woman, it's possible to treat them with sulfonylureas to close their KATP channels and switch them back on again. And it's not just in neonatal diabetes that the beta cells remain functional, because we know that in bariatric surgery, there's a dramatic reduction in diabetes incidence and in the severity of the disease in about 70% of cases. And this is where you bypass much of the stomach so that the food simply shoots straight through. And I just love this little piece that came from Science, which describes the first one of the first surgeons to do this gastric bypass, and he noticed that the patient with type 2 diabetes had an improvement in their disease, and he decided this can't be right. So they went downstairs to make sure that the minions were actually measuring the blood sugar levels, because, as he said, if you're a doctor, you like to blame other people. Couldn't resist putting that in. And sorry, Andrew. I would also like to point out that by improving the blood glucose level with glibenclamide, we can actually reverse some of the damage that very high, uncontrolled blood sugar levels have produced. So this is in our mice, where we turn on the mutant gene at this point here. You can see that the blood sugar level rises very rapidly, and it remains this way. The mouse is diabetic for 4 weeks, and then at this point, we implant, we treat them with sulfonylureas, and you can see that their blood sugar level is beautifully controlled. And this is what their pancreatic islets look like. So here are the control, and this is insulin staining, and you can see that they stain very beautifully for insulin. And there's also a thin layer around the outside of the insulin cells of cells that stain for glucagon. This is a hormone which has the opposite effect to insulin. It increases blood glucose levels, and these cells, known as alpha cells, are found on the periphery of the islet in the mouse. After 4 weeks of diabetes, most of the insulin staining has been lost. We don't know whether that's because, and you'll notice that now the cells all express glucagon. And so the question is, what's going on? Are the beta cells dying off and being replaced with alpha cells, or are the beta cells in some strange way turning into cells that release glucagon? But what I really want to show you is what's shown over here, because so the mice that had 4 weeks of diabetes were then given 4 weeks of sulfonylurea therapy, and just look what happens to my astonishment. Their islets completely recover, so now they are expressing insulin beautifully, and they have reduced the glucagon expression. So I think there is good news here, but there's just an enormous amount of work to do. And this example here may be why, when patients with neonatal diabetes are first treated with sulfonylureas, they require a very high dose of drug, but it declines over time. It may be that their beta cells were somewhat altered by having poor glucose control before, and when glucose control is improved, then they recover. So I've given you the sort of 20-year odyssey about neonatal diabetes and when we're going next. It sounds like a long time, 30 years probably it should say now, but actually it was incredibly fast, and we were very lucky to be so fast, and the reason it happened was because we knew what the gene did in the beta cell. We already had a drug that selectively blocked it and didn't have any significant side effects. It was already in clinical use, so no clinical trials were necessary. So it's to find drugs which are going to, do this in other cases for other genetic diseases or for type 2 diabetes, much, much more difficult, I think, as it's also going to be more difficult in terms of the neurological problems. So a few patients, quite a lot, but not very many compared to type 2 diabetes, have been helped. But although it's only a few, it's a huge benefit for the individual, as I hope I've shown you. And what I hope I've shown you is that there's value in studying basic science, and there's also huge value in studying the rare diseases. And of course, this wasn't something which I did on my own. I was merely one of the conductors of this wonderful orchestra. These are all the people who worked in my lab on this problem, and these are all the people whose work I showed you in this talk. And they are the talented ones and the ones who did all the work, and I'm enormously grateful to them. I'm just the mouthpiece. And there are many marvellous collaborators all over the world who've helped us in multiple ways, but I really just like to mention 3 of them for special attention. Patrick, who has worked with me for 30 years now on understanding how pancreatic beta cells work. Mark, who's helped us enormously with all the modelling and structural studies we've done. And Andrew, who has been a fantastic collaborator without whom none of this story would be telling you today, because it was he who sorted all the clinical side of it, since he's both a clinician and a clinical geneticist. So I want to thank all of them very much indeed. I want to thank the funding bodies. I want to thank the patients for working with us, and also for allowing me to tell you their stories. And I particularly, so this is the first person in the world, I believe, to be treated with sulfonylureas, and this is the first person in the United States. And she's such a poppet. And it's been such an enormous privilege to me as a scientist to be able to see our work translated into clinical practice. It's not something that you ever anticipate. You always imagine that far in the future it might do some good. And you always say that in your grant application, of course, but you never truly believe it will really happen. And I have been enormously lucky. And I've been even more lucky because I've actually managed to meet some of the patients. And this was a meeting that we held, Andrew and I, almost exactly 3 years ago, at the Royal Society, where we were able to bring the children and the families together so that they could meet one another and we could meet them. And I think this is my favourite picture of the whole meeting, of 2 young children, and in the background, the Royal Society's mace. And I've never forgotten somebody coming in and saying, "What are all these children doing at the Royal Society?" It's not, they weren't quite sure if they approved, I have to say. So thank you very much for listening. Yes, of course, if they want to.
Fran would be happy to take questions at this point.
If you've got any.
If you have a question, put your hand up and then wait for the microphone to appear.
No questions, good, we could just go for drinks.
Last chance. No questions. Trouble is, they're all experts in the audience. So I'd like to thank Fran for a fantastic talk, because the Royal Society has different sorts of awards, medals and awards and prizes, and some of them require the recipient to give a lecture. And one has to be very careful in who you choose for the awards that have lectures, because some very brilliant people cannot address an audience, let alone engage them for a full hour. So Fran was an obvious choice for this award when she was nominated. I think she's proved that the awards panel was right to make this choice. I think it's been absolutely wonderful to listen to her. The hour's gone very fast, and we've learned, well, I've learned a lot, and she's covered a huge amount of ground as well. So, Fran, thank you very much indeed. And now you have to be given something.
Oh, I
can
do.
find them. Yeah.
The loot.
Here is your another certificate.
Thank you very much.
And I always have to have a look at these. Ah, right. That's the Croonian Medal,
Thank you.
and I think I might actually keep this, but I won't. This is a cheque.
Oh, thank you very much. Well, it'll be made out to me, so it won't do you any good, I'm afraid. Okay. Thank you so much.